The regulatory principles for approval of biosimilars is very well thought of, and it's a very stringent process, and it is based on an extensive comparability exercise, first of all at the quality level of the protein, of the active substance of the proposed biosimilar compared to the reference product that should be authorized, for instance, when we're dealing with the European medicines in the European Medicines Agency. So the basic concept is that you start from a thorough comparison at the quality level, physicochemical structure, the in vitro functional properties, the structural properties of the protein, and you compare this with those of the reference protein. And if you can demonstrate that your process is very consistent, that the quality of your product is very much comparable to that of the reference product, then you will be allowed to have a much lesser non-clinical and clinical evaluation to get approval for your biosimilar product. And the main message when dealing with approved biosimilars is that they are as good as any other approved medicine, basically. The overall principle of the approach for approval of biosimilars worldwide in those countries and in those regions which do have a stringent regulatory approval process is overall the same. It's all about comparison of the quality, which is the main foundation of biosimilar development, and then the confirmation on the clinical level. Well, it's such that this is also a scientific approach, the production of any biological molecule, and is also subject to evolutionary processes within the techniques that are being used. So even an existing molecule or a drug that has been used for many years is also subjected to novel techniques, to improvement of the process. So this is a continuously evolving situation. It's not because of a biosimilar that the techniques are being improved. This is just a general phenomenon.